Hello students, today we are going to talk about the fascinating field of protein microarrays. If you remember from the previous lecture, we discussed about different types of traditional methods which have been used for studying the protein-protein interactions. Though many interactions have been discovered by using yeast to hybrid and immunoprecipitation study, there has been reports for high false positive rate which poses a challenge on these technologies. Aside from these technical issues, both of these methods, immunoprecipitation and yeast to hybrids, are primarily the endpoint assay, where the whole assay occurs in a closed system inside the cells. So, modulating the experimental conditions and different type of parameters becomes very challenging. The field of protein microarrays addresses some of these limitations, such as providing the open system that enables monitoring the effect of various types of modifications. In today's lecture, we will discuss some of the high throughput approaches for studying the protein interactions using different type of protein microarray platforms. I will provide you an introduction into the powerful protein microarray platform, discuss various types of protein microarrays, understand the basic workflow of any given protein array experiment, and finally, we will go over the basic steps involved in data processing of a protein microarray experiment. The high throughput genomic and proteomic projects are so called because they capture data at the scale of entire organism and incorporating data into relational databases from which insight into various biological systems, organization of physiological networks can be derived. Different types of hypotheses can be made based on these large data sets. The genomic era has fostered the development of many new methods such as sequencing, SNPs as well as generation of DNA microarrays. The success of DNA microarrays at the time when most of the genes were sequenced, it was almost in the year 2000 and 2003 when we had availability of all the gene sequences. At the time, DNA array technology reached to its maximum potential because it was very easy to screen thousands of genes and full genome of any organism such as human, for which almost 20,000 genes were already available. So, by using the DNA microarrays, scientists have shown the potential of high throughput genomic technologies. The success of genomic technologies such as the DNA microarrays have motivated the development of protein microarrays. I think the good idea to add here that genomic is definitely advanced and is leading the way, and proteomic technologies are trying to follow the same path. In this light, scientists thought whether we could also repeat the success of DNA microarrays at the protein array level. So, what are protein microarrays? Protein microarrays are microscopic arrays which comprise thousands of discrete proteins. Now, the concept of microarrays have stirred a great deal of excitement in the proteomics community because it can be applied for several applications such as biomarker discovery, protein-protein interactions, functional characterization of different proteins, identification of different substrates, drug inhibitor studies, and all of them is possible in high throughput manner. So, once the protein microarray technology is fully realized, it promises to enable the study of broad variety of protein features at an unprecedented pace and scale. The protein microarrays fall into two general broad classes, the antibody arrays and test protein arrays. As shown in the slide, the antibody array is an abundance based method which is intended to inform the investigators how much of each protein is present in each sample, or to identify the proteins whose abundance is differentially expressed in one sample as compared to the other sample. There is always a test control kind of study where you want to compare the differential expression of the proteins. So, antibody arrays, they print thousands of antibodies on the chip surface, and it has been used to measure the proteins or the biomolecules in different samples to compare control versus experimental conditions and looking at their abundance or protein expression. In the test protein arrays, the proteins are spotted, not the antibodies, as we talked in the earlier case. This is done by purifying the proteins first and then printing them on the chip surface. The goal of these test protein arrays is to perform functional studies, so that different type of functions could be assigned, different type of biological questions which are related to the protein activities and its functions can be studied using test protein arrays. So, these have been used for assaying the protein function, protein interactions, studying small molecular interactions as well as identification of substrates. As compared to the DNA microarrays, which have shown its promises and potential in various biological applications, there are relatively few studies published using protein microarrays. But over the past few years, there has been many studies, many promising studies which have shown the potential of using protein microarrays for various biological and clinical applications. The protein microarrays still remains very challenging, just because the way of generating the content, the protein, is very challenging. I am sure you agree that even purifying a single protein in the lab setting is not easy. And if you think about protein purification for thousands of protein, that is of course going to be very challenging. So, scientist Gavin Macbeth at Harvard first demonstrated the feasibility of printing the proteins on the chip surface in a high density array similar to the DNA microarrays. In 2000, at the time when DNA microarray technologies had started showing its potential, Gavin Macbeth published his work in Science and showed that proteins could be printed on the chip very similar to what is being done in the DNA array technologies. So, while he was able to first time demonstrate the utility of protein arrays, and protein arrays could be made like DNA arrays, but still he was able to only spot two proteins, one in the large number and one just to show the specificity of the assay, which also opened up the discussion that how challenging it will be to print thousands of proteins on the array, especially if you have to purify all of those proteins separately and then use those for the printing the chip. So, protein content generation definitely is one of the major challenges behind the success of protein microarray field. The success of Gavin Macbeth's study motivated other scientists also to start doing the protein microarrays based research. Dr. Mike Schneider's lab, which is also pioneer in the protein microarray field, they showed that whole yeast proteome could be printed on the chip by purifying 5800 yeast clones, which were histidine tagged, and then they also showed that novel calmodulin and lipid binding proteins could be identified using this novel whole yeast proteome arrays. This was a full scale yeast protein array showed in 2001. The potential of these chip technologies for protein interactions and different type of other functional applications, I must say this was kind of a beginning of the very exciting field of protein microarrays, when many investigators who had access to the large number of clones or access to the purified protein, they started thinking about using the miniaturized platform start printing the proteins on the chip and then performing some very interesting biochemistry at a very small and miniaturized scale. But of course, the throughput will be very large, because from the very small volume of the sample you can now investigate the thousands of proteins which are printed on the chip surface. So, there are different type of platforms which are available for studying the proteins using protein microarrays. Let us have a quick look on some of these available platforms. Let me first start with the abundance based arrays, specifically the use of antibodies for making protein arrays. So, antibodies have been used to print on the chip surface for various proteomics applications in different type of orientations. The ones shown here in the slide show the direct labeling, where the target proteins are labeled with fluorescence or other tag, which allows detection after it is captured by the antibody immobilized on the array surface. What is shown here is the sandwich immunoassay, in which the target protein is captured by an antibody followed by detection with labeled secondary antibody. In the reverse phase protein arrays, the complex mixture such as the cell lysate or the tissue lysate is printed and are probed with the specific detection labels. All of these three methods, direct labeling, sandwich immunoassay and reverse phase protein arrays, they rely on antibodies. While antibodies and antigen interactions are very strong and gives very easy way of doing biochemical based assay, very strong biochemistry can be done if you have access to good antibodies. But access of good antibodies, especially both the quality and the cost in the large number, is definitely a challenge. Therefore, scientists have started exploring different methods of printing protein on the chip surface for different types of applications. The very conventional or most widely used method for printing the proteins involve chemical linkage. The purified proteins are immobilized on the functionalized glass slide, and it can be used for various applications. If one can purify large number of proteins, then of course, this will be the most ideal system when the purified proteins could be printed, and now you can use it for different type of applications. But because of the challenges involved in doing the protein purification at a scale, this method remains very challenging. So, scientists have thought about more alternative ways of making protein arrays. Peptide fusion tags is another approach. Many times if you are doing mass spectrometry based proteomics, you are specifically identifying only peptide sequences, that some of these peptides which are relevant for a given protein are showing differential expression. So, now you would like to only take those peptide sequences, synthesize peptide fusion tags, and those could be printed on the array platform to make peptide arrays. So, these peptides can be synthesized artificially. There are many company manufacturers or even different labs where these peptides could be synthesized. Then these are fused to the different type of tags like GST or histidine tags, and then spotted on the array platform. Depending on what type of tag you are using, you can also think about different chemistry for measuring those biomolecules and different type of interactions using these peptide arrays. So, while we have discussed about antibody arrays and purified proteins and peptide arrays, however, due to the challenges involved in having good antibodies, purified proteins or peptides, scientists thought about exploring alternative ways to eliminate the protein purification process. Dr. George Lebert's group at the Harvard at that time in 2003-2004, while now they have moved to Arizona State University, but this work was done at Harvard when they developed nucleic acid programmable protein arrays or NAPA, in which the cDNA containing the GST tags are printed on the array surface along with captured an anti-GST antibody. Protein was expressed using cell-free expression system and captured by the antibody. I will of course talk about the cell-free expression systems, NAPA and some of these concepts in much more detail later, but just to give the context here, we are simply trying to take the DNA or the cDNA molecules, adding the machinery which can do transcription and translation on the chip itself and then synthesizing the protein directly. This was very revolutionary concept, and of course, the work was published in Science in 2004. Another cell-free expression based method which also tried to overcome the limitations of NAPA, tried to print the cDNA along with the in vitro transcription translation mix, which is a process known as multiple spotting technique or MIST. So, MIST involves cell-free expression, in situ expression of the unpurified PCR products, and the cell-free lysates are printed on top of the spot, so that both in vitro transcription and translation can be performed in the chip surface. So, I hope I have given you a glimpse that there are different type of microarray platforms are available, both using the purified proteins or even antibodies, or one could also utilize even cell-free expression based systems and make use of the cDNA or even unpurified PCR products. Broadly, we can divide all of these things into two different type of protein microarray based system. One is abundance based arrays, one is function based arrays. We discussed about use of antibodies for direct labeling, sandwich immunoassays, reverse phase arrays in abundance based method, and then we talked about chemically linked protein printing, peptide fusions and nucleic acid programmable protein arrays and multiple spotting techniques in function based protein microarrays. I hope now you got a sense that protein microarray field is very vast. There are many ways of printing and measuring the proteins and utilizing this high throughput platform to study your biology and interesting biological questions. Protein microarrays have provided the high density, high throughput platform, which was one of the major achievements of this technology. The very small volume of clinical or biological sample, which is usually the challenge, or the drug molecule, the pharmaceutical products can be used on these array surfaces and multifunctional assays can be performed. However, there are limitations and challenges if using the protein microarray, which includes generating the protein contents, protein purification, how long we can store the proteins, keep it functional, whether they are going to degrade over the time period, the printing quality, whether that is going to get affected. So, many of these things are still the major challenges and roadblocks in the success of protein microarrays. development of protein microarrays, on which thousands of discrete proteins are printed at high spatial density, definitely offers a novel tool to interrogate the protein function in high throughput manner. In this animation, I will discuss different type of features, different type of processes in the protein microarrays and the need for protein microarrays. Functional analysis of proteins is a time consuming process that requires many steps. Analysis of a single protein at a time would be a tedious and laborious process. Analysis of several protein samples will undoubtedly take a long time if they are analyzed at a time. Protein microarrays have successfully overcome this hurdle by allowing analysis of several samples simultaneously. How to express the proteins and purify. The gene coding for the protein of interest is expressed in a suitable heterologous system such as E. coli by means of expression vectors like plasmids. The host cell machinery is used for transcription and translation, which results in a mixture of proteins consisting of the target protein along with other host proteins. Since the protein of interest is expressed along with other proteins native to the host, it is essential to purify the target protein before it can be used for protein microarray applications. This can be done by chromatographic procedures to obtain the pure target protein. Protein purity is tested on SDS based gels. Tags such as histidine 6 are often fused with the protein of interest to facilitate the purification process due to its specific affinity towards nickel. The array functionalization. The array surface is functionalized with a suitable chemical reagent that will react with groups present on the protein surface. Aldehyde and silane derivatizations are commonly used as they interact well with amino groups present on the protein surface, resulting in firm capture of the proteins. Robotic array printing. The printing solution is printed onto the array surface in extremely small volumes by means of a robotic printing device that has small pins attached to it for this purpose. The slides are kept for a suitable duration following the printing steps to allow capture of the protein onto the array surface. The unreacted sites are then quenched by a blocking solution such as BSA, which also prevents any non-specific protein binding in subsequent steps. Types of arrays. There are two types of protein arrays that are commonly used. In forward phase arrays, the analyte of interest such as an antibody or aptamer is bound to the array surface and then probed by the test lysate, which may contain the antigen of interest. In reverse phase arrays, however, the test cellular lysate is immobilized on the array surface and then probed using detection antibodies specific to the targets of interest. Protein detection and analysis. In the direct labeling detection techniques, all the target proteins are labeled with a fluorescent or radioactive tag that facilitates easy detection upon binding to the immobilized capture antibody on the array surface. In the sandwich assay, however, a fluorescent tagged secondary antibody that recognizes a different epitope on the target antigen binds to it and is detected by means of fluorescence. The protein microarray is then scanned in a microarray scanner that allows detection of the fluorescently labeled proteins or antibodies. The output from this scanner is then received by an appropriate software on which the data can be analyzed. Certain well characterized proteins can be printed on the arrays as shown in the animation. Now a proof of concept array is shown here, where well characterized proteins are printed on the array surface along with their corresponding query molecules shown on the left labeled with different fluorescent dyes. Now by using these interactions, let us match the protein interacting pairs Jun and Phos, P53 and MDM2 by dragging the query to the correct protein on the array surface in order to see the signal output. So on the array surface, there are both P53 and Phos proteins present. Now if we drag the Jun protein, it should interact with the Phos protein. As you can see by the interactions here, MDM2 protein interacts with P53 protein. Once the interaction is established, then these signals can be detected using a microarray scanner. So in summary, today you got a glimpse of different type of features which can be printed on the array surface to make protein microarrays such as antibodies, purified proteins, peptides, cDNA or even PCR products by using the cell-free expression system. We also got a glimpse of the history of the development of protein array field, which really got motivation from the DNA arrays. The entire workflow involves very high throughput robotic ways of doing the work, where large number of features has to be generated, they have to be then printed on the chip, and once you have made these arrays, then with very small volume of the samples, now you can test their antibody response or protein interactions, the substrate identification or various type of interesting biological questions could be probed using these arrays in very high throughput manner. In the next lecture, we will talk more about the different type of cell free expression based protein microarray platforms. Thank you.